would like to start this webinar by introducing the coordinators of the program of the international track of the doctoral program in biomedical sciences, myself, Lisette Layton, and Dr. Han Moshak for the University of Groningen. This webinar has been organized by the Joint Degree Program Committee to promote collaborations between the academics and show such collaborations to the PhD students from both University of Chile and University of Groningen, to offer a unique opportunity to connect, learn, and explore different research avenues. Today's topic is aging, and our experts are Dr. Marco de Maria from University of Groningen and Dr. Mónica Cáceres from University of Chile. They will give you a quick glance into the fascinating mechanisms of aging, sharing their research and insights for future collaboration within this program. So each speaker has 20 minutes to talk about their research, while attendees should send their questions through the chat. We will address those questions to the speakers after both presentations. People in the audience can also raise their hand to ask questions directly to the speakers. And the total time estimated is an hour. So I will start by introducing our first speaker, Dr. Mónica Cáceres. Mónica is an associate professor at the University of Chile, and she did all her studies at the University of Chile. She is an engineer in molecular biotechnology. 2002, she received her title and holds a PhD degree in pharmacology since 2011. She was appointed by the Faculty of Medicine, University of Chile in 2013 as an assistant professor, was promoted to associate professor in 2021, and she directs the Aging and Tissue Repair Laboratory at the moment at the Institute of Biomedical Sciences in the Faculty of Medicine. Currently, she is the site director of the Institute. So with that introduction, I'll let you start, Mónica. Thank you very much.
Thank you very much for the invitation. Yes. Okay, hello everyone. Today I'm going to talk about aging, senescence, and inflammation. Nowadays, people are healthy until they get sick for something, and they are treated for that disease. But now we know that most of these diseases are related to aging. Aging is characterized by a decrease in the organ functionality. And interesting, when we're getting old, more than one organ fails at the same time. So in this time, we thought that blood connects all the organs, so we start to analyze blood samples from different age to find the first signs of aging. To do this, we incubate human gingival fibroblasts with blood serum from young people, that's 18 to 22 years old, middle age from 30 to 48, and aging over 50 years old. And we analyze one cellular response related to wound healing, that is proliferation. So we only use healthy people. All of these people is people from the Faculty of Medicine. They are undergraduate student, PhD student, professor. So it's healthy people, functionality. So we analyze cell proliferation. We took the young fibroblasts, and after 3 days, we evaluate a marker for proliferation. And what we found is like when we use blood serum from young people, we have an increase of cell proliferation. And we didn't find this increase in blood sample from 30 years old and over 50. Then we do flow cytometry to evaluate the same response related to DNA content, and we have the same result. Young people have more DNA if you compare with middle age and aged samples. So in that case, we thought that the first sign of aging was around 40 years old, but the cellular response was before. And in that moment, we thought that maybe the blood samples could induce cellular senescence, and that's why we have a decrease in cell proliferation with the samples over 30 years old. When we talk about cellular senescence, this is large cells with an increase of cell cycle inhibitor, that's P16 or P21. Also, they have marker for the liposome related to the beta-gal associated to the senescence, an increase of lipofuscin, and most of the markers for cellular senescence are related with the nuclei. So we can have DNA damage response marker, we have heterochromatin foci related to senescence, to telomere. Also, one of the main characteristics of this cellular senescence is they secrete growth factors and pro-inflammatory cytokines. And so that was the characteristic of cellular senescence. And what we evaluate, we took the young gingival fibrillas with the blood serum and we analyzed senescence. And what we found, that when we use blood serum for 30 years old, we have these enlarged cells with the marker for beta-gal associated to senescence, and also DNA damage response. So in this moment, we thought that the blood serum from 30 years old could induce cellular senescence. And we wanted to know what is the difference in the blood sample that can induce senescence. We did a protein profile array, and then we analyzed some of the candidates by ELISA. And what we found, that most of the chain between middle age and age was related to pro-inflammatory cytokines. And also, we found a decrease in growth factor related to proliferation or angiogenesis, but not, for example, in the case for TGF-beta. Also, another candidate that we evaluate was related to metalloproteinase. This is TIM1. And interesting, this is one of the targets for when we predict biological aging related to chronological. This is one of the candidates to evaluate. And also, we found this candidate modulate during aging in the blood samples. So after all this, the result, we start to think that middle age are the most attractive target for therapies, because you don't have the disease, but eventually you will develop an age-related disease. Also, if we saw that during aging we have an increase of pro-inflammatory cytokines. This year we analyze in the same context how inflamed you arrive and as emergency surgery could predict mortality. So inflammation is very relevant in this case. But we analyze the ratio between neutrophil and lymphocyte. And what we found, people that survived after 180 days after the surgery have low levels or this ratio between neutrophil and lymphocyte. Usually when you're normal, you have between 2. In this case, we have between 5, and the one that disease, they have 10 or even more. And interesting, we can this ratio can predict when you are under 65 years old. Over 65 years old, you cannot predict mortality. So how inflamed you arrive at that surgery is very important to predict mortality. So now we know that during aging, you have an increase of cytokines, and also the ratio between innate and adaptive response are related to mortality. But where it came from, these pro-inflammatory cytokines, we believe that they are related with senescence, because during aging, it has been described that we accumulate cellular cells or senescent cells. But these models are mainly in mouse. So we did it in humans. And interesting, for example, this cytokine is the one that we evaluate in blood serum. So some of the cytokines that we evaluate in the blood serum are also secreted by a senescent fibroblast. So how we start to see if during aging we accumulate cellular senescent in human gingival tissue. So the first thing that we evaluate was the gamma phosphorylation of the histone 2. That's a marker for DNA damage response. And we found that during aging, there is an increase of the percentage of the cells for this marker. And also the level of deposited cells for this was very similar to the one that we observed in very old mice, because we found like 20% of the cells have this marker. And also we evaluate another marker that's related to heterochromatin foci, and we can observe that the young cells have the heterochromatin in very close to the border of the nuclei. This is, you can find also this in very proliferative cells. But when we are getting old, we can start to see this marker for heterochromatin modification in the middle. That's related to the senescence-associated heterochromatin foci. So it's very, if you compare, so what we analyze in more than 30 samples, the distance for the marker to the peripheral of the nuclei. And what we found, that during aging, we have more of this foci if you compare with the young cells. Also, we analyze the typical marker for senescence that is related to cell cycle, that is P16. And also during aging, we have an increase of these cells that correlate with the increase of the DNA damage response, that is 53 BP1 marker. So we have 3 markers related to aging, and especially for senescence, that increases with aging in human gingival samples. And then we start to cultivate the gingival fibroblast. And what we found, that when we look for the morphology of the cells, we can observe that the young cells have a very small nuclei if you compare with the age one. This is like, for example, 56, 58, and the nuclei look very different. So we start to quantify different nuclear aberration that could be modulated during aging, because we already found the tissue, and now we're cultivating the human gingival fibroblast. And what we found, that during aging we have bigger cell nuclei with more area, and also they lose the form like an ellipse that is very canonical for the fibroblast. We also quantified nuclear invagination for lamine B1. This is also very, very described in progeria models, an increase of this invagination, and we have the same in our cells. And also we quantify a marker for MAP kinase stress, that's 44P38, and there is a relationship between the form of the nuclei and the marker for the stress. We can observe, for in this case, the gray one are the young one, and the pink one are the cells that belong for an age donor. And also the age donor are more dispersed if you compare with the young cells. In this case, we wanted to know, why do accumulate senescent cells? And we did a model, but what we believe, the cells that accumulate is because the immune system do not eliminate that. So in this case, we use a marker for senescent cells from young and aged donors, and we incubate with PBMC to see if the cells are different to attract and to be eliminated. So in that case, we can observe that the young senescent cells is able to recruit more cells than another senescent cells that became for an aged donor. So even when both cells are senescent, the one that belong for a young donor is able to recruit more cells than unaged senescent cells. Why this is important? Because even when both cells are senescent, during aging, we accumulate these cells. So that means that there is something with the cells that became from an old donor that could be like the key for why we accumulate senescent cells. So after this, we quantify. And the same that we saw at the video, when you have a senescent cells from young donors, the first cells became faster. They arrive more cells if you compare with old cells. So both cells have the marker for senescent cells, but they are different. We don't know why, but we know that the senescent cells that became for a young donor, you can eliminate faster than the old one. Also, we quantify when the first time arrive, if you compare with the young one, they arrive on cells, then another, and they became all the time. And if we observe for the old cells, the cells arrive, they stay for a time, and they leave. And we analyze several videos for this. So we have that the old cells is able to recruit less cells if you compare with the young one. But also we wanted to know if the cells can be eliminated. So we did the same co-culture model, but in this case, we use lysotracker for the PBMC. That means if the PBMC is going to eliminate the senescent cells, will be between the acidic granules that contain granzine and perforins. That's the main mechanism involved in cytotoxicity. So what we observe after 4 hours, we can observe in the human gingival fibroblasts the marker that was in the PBMC. So we quantify this. How many of the senescent cells are positive for the lysotracker that was in the immune cells? And what we observe, that the young cells, the young senescent cells, recruit more cells and also have the marker for the PBMC. That means that the senescent cell for the young cell is going to be eliminated faster, or at least have the transfer for the immune cells to the human gingival cells. So in this case, we know that the cells are different, and we're worried about the senescent cells that persist on the time, the one that we have when we are getting old. So in this context, we found a protease, a serine protease, that the expression is modulated during aging. So that means that in an aged tissue, we have an overexpression of this protein, the same we found in rat and mouse. So this is a very good candidate because it's a protein that is modulated during aging and is related to senescence. So we found that the knockout of these cells, or these protease, have less marker of the cellular senescence. And in a HEC model, when you overexpress this protease, we have more cellular senescence. The same happened with the DNA damage response. The knockout have less of this marker if you compare with the wild type. And interesting, the cell cycle inhibitor is P15 and not the canonical P16. But also these cells have problem to proliferate. So what we did, we took an old mice and we used lentivirus with the small harpin against the protease. We wait 3 days for the virus, and then we did a wound healing assay. When we found that the attenuation of this protease increased wound healing. So we have a smaller wound area. And interesting, what we found, and when you attenuate the expression of this protease, cellular senescence in the old mice decreases. So this is why we are studying how this protease interact with other protein and how it induces senescence. Because we are, as we saw with the human part, the senescence cell for a young donor is able to be eliminated, but the one for the old one is not that easy to be eliminated. And in this case, we found that in an old mice, when you attenuate this protease, you can decrease cellular senescence in an old mice. That's the one that we are trying to eliminate, because the problem with the senescence is when the senescent cells persist on the time. So what we're trying to do, we have some candidate. I'm not going to show this result, because some candidate that interact the protease with ligand that are related to the elimination related to the immune system, the interaction with the natural killer cells. And that's it. I would like to thank all the collaboration, because we saw that we have human samples, so we have an MD, dentistry, we have people that help with.
Thank you, Monica.
Thank you.
The questions will come at the end, I think, so we will continue now with the other seminar. So if Han can introduce our next speaker, please.
Yes, thank you very much. The next speaker is Professor Marco de Maria from the University Medical Center in Groningen. Professor de Maria studied molecular medicine at the University of Turin in northern Italy. And after his PhD, he continued to do a postdoc in the well-known Buck Institute on research on aging in California in the United States. In 2010 it was. And in 2015 he joined the University Medical Center Groningen in the European Research Institute for the Biology of Aging, where he is group leader and professor in cellular senescence and age-related pathologies. In addition to this, he also is the co-founder of a spin-off company called Clera Biotech and editor-in-chief of the journal Aging and president of the International Cell Senescence Association. And it's a great pleasure for me to ask Professor de Maria to take the floor and present his seminar.
Hey, thank you very much, Han, for nice and kind words, and also for the invitation to be here. I'm happy to talk to you about something that is ongoing in the laboratory regarding our research on cellular senescence and its involvement in aging. So good afternoon, everybody, of course, from the Dutch side, and good morning, everybody, from the Chilean side. I was actually in Chile just in November for a wonderful meeting of the Cell Senescence Association that actually Monica organized. So it is a pleasure to be here, part of this collaborative effort between the RUC and University of Chile. And I hope that we can actually at some point continue this effort, and I can also have somebody in the lab coming from Chile. So Monica already touched upon a few things that I wanted to introduce to you, but I like to give a little bit of an helicopter view of the role of senescence in aging, starting from the idea, as we mentioned already, it was mentioned already by Monica, that aging is the predisposition to virtually every chronic disease that we can think of. Here you see the major diseases that we associate with chronic disabilities, including cancer, dementia, diabetes, and many others, which all exponentially increase with age. But I think that the major issue that we have in the society right now is the concept of multimorbidity, meaning that we are not only looking at individuals developing one disease, but we're looking at individuals developing multiple diseases at the same time. And the older we are, and the more disease in the same individual we can detect. This poses a very important question on how we have to approach it from a pharmacological point of view. Because in medicine, what we are used to do is to try to target individual diseases. First of all, as now we think about multimorbidity, this might become a problem because, of course, we are now trying to target different individual diseases at the same time with different drugs, which might have, for example, interactions, antagonistic interactions, which might give each of them some side effects. And second point is that most of the drugs we have that are targeting chronic diseases are not able to cure anything. They normally just delay the progression of the targeted disease. So what we are currently doing in medicine is essentially sick care. So we're keeping people in a sick state, and we're trying to keep them in a decent state, but the quality of life of these individuals remained very poor until death. What we're trying to do in opposition to this type of approach is to try to understand whether there are certain basic mechanisms of aging that might predispose us to develop this multimorbidity process, which, if targeted, could eventually prevent the onset and progression of multiple age-related diseases at the same time. This is the real concept of healthcare, because in this case, we are really trying to take care of the healthy individual and trying to maintain and to extend the period of health that we have. So, of course, this requires that we know or we understand these basic mechanisms of aging. In the last decade or so, a lot of effort have been put in trying to indeed identify these hallmarks of aging. So this is a representation, a very famous one, published originally in 2013, and then in a second version in just last year, actually in 2023, by Carlos Lopez-Otin, Manuel Serrano, and others, where these potential basic mechanisms of aging are represented. Now we have these 12, which have some sort of different hierarchies in how they happen, but essentially we can detect and identify these mechanisms of aging in essentially every type of older organism, at least higher organisms. And among those, as we of course want to discuss today, is the concept of cellular senescence, which was already introduced by Monica. Senescent cells are cells that are, as main feature, cells that lose irreversibly their ability to proliferate. And this is because of the upregulation, for a way or another, of two important pathways, the P16 RB pathway and the P53 P21 pathway, which both are in charge to essentially lock cell cycle progression, mainly at the G1S phase. But in addition to this cell cycle arrest, senescent cells are characterized by multiple morphological, functional, and structural changes. You see here some examples. I'm not going to focus on any of those in particular, but I will just focus on one of these. It is important to know that this state of senescence can be triggered by a number of different stresses. We go from telomere shortening, which was the first description on how cells age and become senescent, to other type of inducers and stresses, including metabolic dysregulation, aneuploidy, excessive DNA damage, epigenetic alteration, mitochondrial dysfunction, oncogene activation, and excessive oxidative stress. So all these can essentially push cells that normally would be able to proliferate and to give rise to progeny to become senescent, to remain metabolically active, but then to indeed lose the ability to proliferate. And one of the major features of senescence, which I wanted to focus on, is the SASP, so the senescence-associated secretory phenotype. So the SASP, as mentioned by Monica before, can include a number of proteins, for example, interleukins, growth factors, metalloproteases, and others. But we've also learned in the last few years that the SASP is much more complicated and not just a program that includes secreted proteins. There are indeed among these program lipids, there are extracellular vesicles, and then there are various nucleotides that can be, for example, free DNA, circular RNAs, and others. And all these factors act either autocrinally by reinforcing the senescent state or paracrinally by modulating the behavior of the neighboring cells. And the SASP, because it's so heterogeneous and also so complex, has a number of biological functions. And broadly, we can divide into what are the beneficial and what are the detrimental function of the SASP. On the beneficial side, we know that senescent cells, in particular when they are transiently present in tissues, they can stop the propagation of tumor cells. Of course, the cycle arrest is a feature that would counteract the harm of being cancerous cells, which normally are characterized by unrestrained proliferation. They can induce senescence to the paracrine tissue, meaning that they can expand senescence to other cancer cells, but also they can induce the death of certain cancer cells, so acting as very potent tumor suppressive mechanism. They can engage into immunosurveillance of the cancer cells themselves, so further supporting this tumor suppressive mechanism. But they also participate in various tissue remodeling and tissue regeneration processes, both during embryogenesis and adulthood. In contrast to this, especially when senescent cells persist, they become detrimental, and they start to disrupt normal tissue homeostasis. They paradoxically seems to promote tumor progression and immune cell evasion. And they start to create what also Monica mentioned, which is a low-grade chronic inflammation, which seems to be one of the major factors involved in promoting aging-related pathologies. And one of the issues that we have is that senescent cells seems to naturally accumulate with age. So here you have some examples in humans. These are very different tissues, also from a mitotic perspective. We have the skin, we have the colon mucosa, and we have the brain. In all these cases, without going into the details, what you can see is there is an enrichment in senescence-associated markers when the tissue ages. So you can see in all these tissues, for example, the P16 becomes overexpressed when the tissue is from an individual that is older than 55 years of age. Then you can also repeat the same using a mouse. And here is an example where you take a mouse that report the presence of senescent cells, in this case, because they are labeling essentially P16 positive cells with a luminescent reporter. And here it's a mouse that we have generated a few years ago. It's called P16-3MR, where indeed there is the expression of this reporter gene that encodes a luminescent protein, but also a fluorescent protein and a suicide gene under the regulation of the P16 promoter. And when you follow the same animal over time, so from young to older age, you can clearly see that the luminescent signal exponentially increases with time, suggesting that these P16 positive cells become overexpressed in aged tissues. And interestingly enough, not only this age-associated senescence is accumulating, but is also detrimental. Because now if we use the trick of eliminating senescent cells from an aging mouse, and this is possible because indeed of the suicide gene that we have in this transgenic model, we can clearly see that the mice maintain a period of health for longer term. So if we measure endurance or strength from mice that have been eliminated of senescent cells at older ages, so 21 months of age, they are clearly performing better compared to mice that are still in the presence of accumulating senescent cells. And similar lines, a study from Jan van Deursen and Darren Baker from the Mayo Clinic and in Minnesota reported a few years ago that if you eliminate senescent cells, also because of a transgenic trick, using a different type of model, you actually can also extend longevity in these mice as a good side effect of also living better. So essentially, senescent cells seem to lead to aging in various ways. And we also know that senescent cells become overrepresented in various chronic diseases. So this is an example or some examples of what happens and what type of senescent cells we are able to detect in various types of diseases in humans. And you see that senescent cells seems to be overrepresented in neurodegenerative diseases, in cancer, in fibrotic dysfunctions, in metabolic disorders, you name it. So essentially, virtually every chronic disease seems to be associated to a higher burden of senescent cells. So this made us thinking that perhaps it could be a good strategy to develop interventions that could eliminate the detrimental effects of senescent cells. So what we generally and broadly call xenotherapeutics. So all those type of compounds that would in one way or another interfere with the biological function of senescent cells. And this actually brings us to another important concept, because it's not that easy to find the Achilles heels of senescent cells, because senescent cells are highly heterogeneous. So we have done some studies in the past, but we have found that there are various extrinsic and intrinsic factors that can determine whether and how a cell would develop a senescence phenotype. And all these senescent subsets express different combination of genes and markers. So to eliminate or to trying to interfere with these cells can become highly complicated because, of course, they all have different features. In addition to this, when you take, for example, a mouse, you can clearly see that there is an enormous heterogeneity in how and what type of cells become senescence in each tissue and what type of behavior they have. So, of course, the heterogeneity of senescence needs to be taken into account in order to develop effective xenotherapies. And also, we need to keep in mind that there are various ways to interfere with senescent cells. On one side, what we can think is that perhaps if we find certain shared features of senescence that are at the core of the survival mechanism of senescent cells, we can develop what we call senolytics. So drugs that could selectively eliminate these senescent cells. So selectively induce these cells to die. On the other side, we can also think that if whatever detrimental biological function of senescence is driven by the SASP, we can generate and develop inhibitors of the SASP and then potentially interfere with the detrimental function of senescent cells. So briefly, I give you an example of both. The first one came from a study that we had done a few years ago with a student in my lab, Alejandra, which was very interesting to understand the transcriptional heterogeneity of senescence and to find what we call the core transcriptional signature of senescent cells. So what Alejandra found was that at the heart of and at the core of the senescence signal, when she tried to put different senescence models together, was this very different regulation of apoptotic modulation. So what it looked like was that senescent cells seem to be primed to apoptose, but on the other side, they also overexpress anti-apoptotic proteins. So we reason that if we were to develop or to use anti-apoptotic inhibitors, we would then have the senescent cell to commit spontaneously to apoptose again. And this is indeed what happens. So here you have the example of using one of these compounds. Our purpose compounds is called ABT263 or navitoclax, which is an inhibitor of a family of anti-apoptotic proteins, which is called the BCL2 family of proteins. So when you apply this compound on non-senescent cells, nothing happens. So the cells remain viable and they continue to proliferate. However, when you apply this compound on senescent cells, on different type of models of senescence, you clearly see the viability drops dramatically, suggesting that the senescent cells are now dying. And this is true also when we use this compound in vivo. So here you can see that this is a mouse where you see the luminescence. And then either if you use the genetic trick with our transgenic model, or you apply this ABT263 compound, you see that this luminescent signal essentially disappears, suggesting that also in vivo, we are able to eliminate the cells. And now it is important to note that one analog of the BCL2 inhibitors is in phase 2b trial for the treatment of diabetic macular edema. So we are waiting, and this is spearheaded by a company called Unity Biotechnology in the US, and we're waiting for the release of the data this year, and they seem very promising. So we'll see whether this compound indeed is able to target senescent cells in this condition and also improve health of patients with diabetic macular edema. And it is important to know that we have now learned that there are different types of senolytic approaches, which in a way or another are all working on the same idea of trying to re-engage the apoptotic, anti-apoptotic balance in senescent cells in order to make the senescent cell to spontaneously die without then harming non-senescent counterparts. And then as a second option, as I mentioned, it would be important to consider also some sort of SASP inhibition. And here I wanted to give an example that also started by studying essentially basic mechanisms of senescence and senescence heterogeneity. This work was pioneered by Thymen, a PhD student in the lab. Thymen was extremely interested to understand how different oxygen concentration, which are very different in tissues in our body, could influence the induction of senescence phenotypes. So he used a normoxic environment, which is around 5% oxygen, which is the one that normally our cells within our organs would be exposed to on average. And he also used what we call physiologically hypoxic environment, so 1% of oxygen, which certain part of our bodies, for example, part of the kidney, skin, bones, and others are exposed to. And then he studied how senescent cells evolve in these environments. So he didn't see any particular difference in the entry of into senescence in terms of graft arrest. So both conditions physiological hypoxic or normoxic conditions, were able to make a cell to enter this growth arrest irreversibly when given to a senescence inducer. However, what he measured was a very different level of SASP development. And in particular, Tymon discovered that in hypoxic conditions, senescent cells drop the secretion of many harmful pro-inflammatory SASP factors. So then he reasoned that perhaps using hypoxamimetic compounds, it could achieve the same effect. So now he kept his cells in normal oxygen concentration, but exposed them to an hypoxamimetic compound called roxadustat and measured the SASP. And again, what he could see was that the exposure of senescent cells to this compound was sufficient to drop the pro-inflammatory SASP. So then he went in vivo as well, and he used this compound on aging mice, and he found that also in this case, when looking at tissue SASP, he could decrease the expression of these pro-inflammatory harmful SASP simply by exposing the mice to roxadustat. And also these mice showed improved health. For example, they were stronger than the counterpart that didn't get the compound. And it is important to note that now we have, again, as in the case for senolytics, we have now a number of different SASP inhibitors that can be used to treat at least preclinical preclinical systems, and in particular mouse and rats, some monkeys, and we have some that are now in clinical trials. And indeed, it is important that to close this session by highlighting the fact that many xenotherapies now are in clinical trials. I mentioned one in phase 2b, which is the most advanced, but we have a number that are in phase 1 or early phase 2 with different concepts using different indications. But it is very promising that now senescence is a target for interventions with the potential to improve and extend healthy longevity. So to conclude, I hope that in this brief talk, I could show you that senescent cells accumulate during aging and they contribute to chronic disease and multimorbidity. I also mentioned that the senescence program is very heterogeneous and depend on various intrinsic and extrinsic factors. By studying how the different senescence programs develop, we can also find potentially xenotherapies, so compounds that can counteract the detrimental function of senescent cells. And we have some very interesting evidence and it at the moment, and some of these are now tested in humans. But I also would like to emphasize the fact that more studies on characterizing and targeting different senescent cell subtypes are still needed. So we still need a lot of money and effort to study senescence and the basic biology of senescent cells. So this is my slide of acknowledgements. I mentioned people that contributed to the studies I showed you. And thank you very much for your attention, and happy to take any questions.
Thank you, Dr. De Maria, for your wonderful talk. Let's start then with the questions. We have one question in the chat from Dr. Fabiola Osorio. This is for Mónica Cáceres, Dr. Cáceres. And she says, nice to talk, Mónica. Nice talk. IL-11 is a cytokine recently linked to aging. IL-11 knockout mice exhibit markedly delayed aging features. Have you found differences in IL-11 levels in serum from middle-aged and aged people?
Yeah, I read that question. I know that this interleukin is related to longevity, but when we did that study, we didn't thought about this interleukin. Probably it will be modulated, but we didn't do it. I think it's a great question, but probably it will be modulated.
Okay, thank you, Mónica. There is another question now for Dr. De Maria in the chat from Javiera. Thank you, Dr. De Maria. Have you seen any difference between SASP from senescent cells and the secondary phenotype from cancer cells?
Yeah, I guess the question relates to also a senescent cancer cell. I don't know, but it is the problem is that cancer cells are very difficult to study because they are. If we think that senescence is heterogeneous, cancer is even more. So the SASP is really dependent on various type of mutations that might be in the cell that is indeed transformed. They might be dependent on the environment where the cells sit, you know, depends on which area of the tumor is. So it's very difficult to pinpoint whether there is any similarity in the secretory phenotype of cancer cells versus senescent cells. But for sure, both of them are trying to do something related to immune regulation and also angiogenesis, growth. So there are some common features, especially in terms of like categories of biological functions that are touched by these secreted factors. However, maybe the individual factors might not always be redundant or overlapping.
Okay. Thank you very much. We have more questions for Dr. De Maria. Dr. Claudio Hetz says, excellent talk, Marco. Claudio Hetz here. What do you think about the possible side effects of using BCL2 inhibitors as treatment? In the field of cancer, there are now many side effects described, including altered bone mass and other effects.
Yes, of course. Yeah. Hi, Claudio. Yes, yeah, that's a good point. I mean, indeed, I don't believe that this one will be, you know, the real strategy to use to target senescence. However, I would say that it depends really on the context. So at the moment, the trial on macular edema, which I mentioned, is using a local treatment. So they just inject essentially the compound in the eye. And that might work very well to avoid all these side effects, right? So it's a drug that could be applied to local treatments for sure. For systemic treatment, indeed, I agree that there might be side effects. However, the regimen also and the duration of treatment might be very different between a cancer patient and a patient with some other age-related disease. So we also need to keep that in mind. But indeed, we need to be quite careful about potential side effects. There are some very well reported, neutropenia, and in particular thrombocytopenia, which are driven by BCL2 inhibitors. However, these are kind of transient, but can give some problem to certain patients. So for sure, we need to be careful on this. And as any drug, unfortunately, also these ones are having their own problems. But, you know, especially going with revised versions of these compounds, maybe more specific, more targeted, we might avoid some of these side effects.
I don't know if Han wants to read some of the questions too. There are more questions in the chat.
Yes, I can see that there is another question of Dr. Fabiola Osorio, also to Professor De Maria, to Marco. Is it possible to selectively target senolytics to senescent cells? And related to that, can senolytics influence other immune responses, for example, infections?
Well, yeah, so regarding the first question, there is a lot of interest now to trying to develop these targeted, essentially delivery systems. So put a cargo like a BCL2 inhibitor, but within some sort of delivery system that would be more specific to and targeted to senescent cells. In the beginning, there was a lot of interest around using SA-beta-gal, which is an enzyme expressed in the lysosomes, which activity is upregulated during senescence, and use some sort of like delivery system that would be like a sugar that is degraded by beta-gal specifically in senescent cells, and then it can release the cargo just in senescent cells. So there are some strategies. The other way would be to use, of course, surface proteins that are more specific to senescence. However, that effort has been there for a long time, and including our lab has really tried hard to find something that is selective for senescence, that is expressed on the surface. So then you can use, for example, antibodies or other ways to target. But at the moment, I would say we don't have anything that would be safe to be used. But, you know, these would be the strategies. Well, senolytics can influence for sure, and the immune responses. But on the other side, what I would highlight is that some senolytics might actually work also because they engage the immune system. So as you know, Monica also mentioned before, one way is to try to modulate the immune system to make the senescent cells more easier to get rid of because of the natural immune-mediated clearance. So that's also one way to look at. So trying to, you know, remodulate a little bit the immune response by using the senolytics might also help to clear them. So it might be that they influence immune responses to infection, for example, but just because they might remodulate how the immune system is.
I also have a question related to this that you're talking about. Do senolytics distinguish between a harmful and a beneficial senescent cell?
Sorry, I missed that question.
Whether the senolytic drugs can differentiate between those senescent cells that are harmful or beneficial?
No, absolutely not. And that's a major issue. That's something that we are working on very hard to trying to. It's very difficult to say, well, this feature is only associated to the detrimental senescent cell. There are some, but they're difficult to target. So there is no space selective intervention for the detrimental senescence. That would be, of course, this is one direction we're taking. Yeah.
Can I ask a question related to the previous questions? That is, again, with the cell selectivity of your interventions. I also noticed it in one of your slides, the liver. You have the hepatocytes, the functional liver cells, and the stellate cells, the ones that cause fibrosis. So senescent hepatocytes, they elicit, let's say, it's not good because you lose your liver function. On the other hand, if you induce senescence of stellate cells, it has been hypothesized as a kind of therapy for liver fibrosis. So inducing or preventing senescence in one organ, if you are not selective in which cells you target, might have opposite or conflicting responses. How do you see that for any future therapeutical intervention, for example?
Yeah, no, it's an excellent question. I think that there are two important points here. The first one is that it's also a matter of time, in the sense that a chronic senescent cell is never good. We never found a chronic senescent cell that is good. So if you can induce senescence in a way that the state is only transient, then probably it's going to be good. The second important point is that indeed, when you, for example, clear senescent cells, that's going to be important to know when you do that, because, for example, we know that they are important for tissue repair. So you don't want to be under senolytic therapy when you're going under surgery, for instance, or this type of interventions, right? And as you said, stellate cells indeed, when they are senescent, they seem to help to remodel tissue after injury. So, and this is true also for the skin. It seems to be true for the heart. We have seen that in kidney for ischemia reperfusion models, where ischemic kidneys also need more senescence to heal and recover. Otherwise, they would degenerate quicker. So it's all a matter of time, I think. It's really a matter of when we are giving intervention and for how long. So we need to be very careful. And I think that intermittent treatment with senolytics is the way to go. We don't want to have people that are chronically on senolytics, for sure.
Yeah. Okay. Thank you very much. And then I see in the chat. Yeah, Lisette?
Yeah,
yes?
I have one more question for Monica also. Do you know how different are the gingival fibroblasts with respect to the senescent cells of other tissues?
They are not that different. For example, if you compare with the one for the skin, but it has been described, for example, that collagen phagocytosis is different between the gingival and the skin, because if you have an injury in the gum, we don't develop any scar. So it's just a model to compare. This is like a very successful wound healing, I'll say. Because, for example, when you have the tooth extraction, after one day, you can eat. And that doesn't happen in other tissue. So it's a very faster healing response if you compare with other tissue. But like a fibroblast, they have the same, it have not described too many different with the other one, but they are more resistant. For example, they have very good collagen expression, and they migrate. Maybe they don't describe it, they are faster than the skin fibroblast, but we know that wound healing is better in the gum if you compare with other tissues. They don't know why, but it is better.
Thank you.
And also probably the senolytic should be at middle age. Before that, talking to Marco, what he said, it should be in that age, because when you're getting old, eventually you will develop the age-related disease. So it should be before that.
Okay, thank you very much. We have another question in the chat as well, again from Javier, related to cancer, to Professor De Maria. Is it possible that the immune system activated by the SASP has any effect on the elimination of cancer cells?
Oh, absolutely. I think that's one of the beneficial function of certain type of senescent cells, which seem to indeed being actually recruiting the good immune cells that would go at the tumor site and eliminate directly via cytotoxicity cancer cells. So for sure there is that, and that's also why, again, in the cancer lesion, it's very complicated to target senescent cells because they might have this immunosurveillance feature.
Okay, thank you very much. I don't think we have any additional questions in the chat at this moment, but please feel free to ask questions. I have one question that is also a little bit related to my own research in metabolic diseases, liver diseases. It seems that mTOR is a central regulator more or less of everything. Like in physics, you have a theory of everything, but in cell biology, you have a regulator of everything, which is mTOR, which is of course also related to, let's say, metabolic stress, food deprivation, which is also related to longevity. So would mTOR be a target, for example, to avoid senescence or aging?
Well, so there are different theories on what the role of mTOR in senescence is. So on one side, it seems to regulate this process that we call gyroconversion. So essentially, what it seems is that mTOR decides whether a cell would enter a state of quiescence, so meaning that it would then be able still to re-enter proliferation if given the right nutrients and the right environment, or senescence. So becoming irreversibly growth arrested. And when mTOR is activated, seems to push the senescent side, so to gyroconvert these cells. This is very tricky because that happens very early and only in certain cells and only under certain stresses. What we see is that mTOR for sure is a major regulator of the SASP, or the pro-inflammatory SASP, because he has a huge crosstalk with NF-kappa B, which is of course a very important pro-inflammatory transcription factors, one of the most important pro-inflammatory transcription factors. And we know that targeting mTOR can also reduce the pro-inflammatory SASP. So there is this dual role of, maybe preventing if you use mTOR inhibitors very early on. However, there is also the risk that if you do so, the cells would have some sort of damage, because maybe it's exposed to damage, and then it would not enter senescence, but it would start to proliferate, which of course is big risk of expanding the damage to other cells, and maybe, might be a cancer, but it might be also other, disruption of tissue homeostasis. But on the other side, what we know is that once the cell enters senescence, it uses mTOR and mTOR-related mechanisms to fuel the SASP. So using, for example, rapamycin seems to decrease the production of pro-inflammatory SASP. So it's still there, the debate, and especially now that, of course, rapamycin is a very, very important and popular anti-aging intervention. There is a lot of questions on what's happening actually in people that are now taking rapamycin. I'm not saying for recreational purposes, but they're taking rapamycin without a prescription. So they're just taking it because they're trying to counteract the aging phenotypes. So it's very important to understand whether this potential prevention of senescence can be also a little bit risky. Okay, thank you very much.
Thank you. There are more questions, but I think we are already on the deadline that we'll post for our seminar. So I don't know if there is one important question that is related to senescence and endocytosis process, thinking of the treatment of drug delivery that needs to be endocytosed by the cells. So are there alterations in endocytosis during aging?
For who is this question?
It's open. It's not directed to.
Okay. Because I see that there is a Dr. Mónica Cáceres who would like to answer this question. I have a note there. So Mónica, I leave it to you.
Endocytosis is not my field, but it is modulated during aging. I don't know, especially if it could be for the senescent cells, but probably it should be.
But maybe in those omics that you've done, have you found any RAP proteins altered?
We did, but in other contexts. We were studying another receptor, and they are different. We didn't quantify that. So I know that it should be, but we didn't do it. But they look different. That's true.
Okay. Let's stop here. I think it's been a wonderful seminar, very enlightening discussion. And I hope that all our audience finds the same too. Very stimulating seminar. I would also like to express our gratitude to all members of the Committee of the Joint Degree Program, whose collaboration has made today's seminar possible. Looking ahead, we are excited to invite you to our next webinar, part of a series that we'll host this year, with 2 webinars each semester. These sessions are a great opportunity to hear about cutting-edge topics and engage with experts in the field. So please keep an eye on our website and emails for details of the upcoming webinars. If you would like to hear about a particular topic, please let us know, and we'll keep it in mind to consider the different topics. So thank you once again for your participation today. Have a wonderful day, and we look forward to welcoming you at our next event. I don't know if anybody wants to add anything.
I have nothing to add, but I would like to thank the organizers of the webinar for the excellent work they have done. So,
thank you very much.
Thank you.
Thanks for joining us today.
Good.
Goodbye.
Bye.
Bye.
Thanks a lot, Mónica.
Thank you.